A tiny organelle keeps records beyond your wildest thoughts. Think of the endoplasmic reticulum like a deep-sea pressure gauge, constantly measuring systemic stability. We always thought this cellular machinery only worried about folding proteins, right? But that misses the signal entirely. Its stress response pathways are actually registering fluctuations in how coherent your entire system remains, a subtle biological pulse. It is acting as an integrated sensor array for self-organization itself. This isn't pathology, it's a profound signature of informational baseline maintenance within living matter.
When we see these subtle changes in cellular structure, like shifts in the endoplasmic reticulum shape, think of it less like a malfunction and more like an ancient barometer responding to deep tectonic pressure below. The research indicates that this organelle actively modulates its own form based on systemic rhythms. It's not just monitoring protein folding efficiency, it is registering how stable the entire information network within the body remains. It functions as what some call a somatic repository of ancestral memory, suggesting we are witnessing biology keeping score across timescales far beyond simple cellular life cycles.
But how does that deep score translate? That cellular memory doesn't just sit there silently in its structure, it actively participates in a larger awareness system. Imagine your entire body is one massive, finely tuned orchestra conductor, and cell signaling is its complex sheet music being read across generations. What we see earlier functions as the groundwork for something far more expansive. It relates to what researchers call the primordial substrate of unified awareness itself. This means that subtle informational coherence within a single molecule has ripples stretching towards an akasha-like field, showing how foundational biology maps onto existence itself, and which means something nobody expected for consciousness studies.
But that foundational biology, that subtle signature of coherence within every cell's machinery, how does our current science even account for its persistence? It feels like a quiet echo across epochs. Think about high-frequency sonar mapping deep underwater. We see structural echoes that suggest entire layers of history existed without leaving obvious wreckage. That persistent informational quality suggests the processes are far more robust than simple chemistry permits. Some skeptical studies argue this signal must simply be systemic noise or measurement artifact, yet the consistent pattern is too fine to dismiss. What drives a system towards such fundamental self-organization? We need to look at how these ancient regulatory states inform everything that comes next.
The system doesn't just maintain coherence, it actively filters information itself. Think of its regulatory pathways like an impossibly complex deep space communications array constantly sifting through cosmic background radiation. It discards static, yes, but also registers specific faint signals that defy conventional understanding of cellular mechanics. This filtering action isn't simply maintenance work, it establishes a state of profound systemic calibration. But what kind of information is being prioritized in this high-stakes internal broadcast? We are only monitoring the upstream trigger event, the molecular firing itself. The next stage, where these calibrated signals actually cascade into whole system awareness, that shift fundamentally changes everything we thought biology meant. But once that internal calibration occurs, what happens next? The sheer persistence of those deep regulatory states suggests something far beyond just a cell keeping itself tidy under strain. Imagine tuning an impossible antique radio. You aren't looking for clean station reception. You are listening for the ghost broadcast from 3 different dimensions simultaneously. That's where we pivot. We realize that what looks like simple cellular response is actually a highly sophisticated endogenous sensor array. It isn't registering damage or failure at all. Instead, it measures coherence itself, a baseline stability of system information far wider than anything our usual metrics account for.
The sustained registration of that deep systemic coherence demands a framework beyond just observing internal stability. It requires accounting for how memory persists across vast gulfs of time and organization. Imagine tuning into an extremely stable quantum resonator, where its resonance frequency doesn't reflect immediate conditions, but the cumulative informational load imprinted upon its fundamental structure itself.
That intrinsic capacity to hold data suggests we are mapping something deeper than mere cellular function onto what some call akasha-like fields, the primordial substrate of awareness. This isn't just biology, it hints at an organizing principle for existence. The actionable insight here is this: view any sustained biological rhythm not as a maintenance cycle, but as evidence of informational self-coherence maintaining itself over immense timescales. The full research archive is at jmthomasofficial.com.
What does that deep informational coherence really mean for us now? It suggests something profound, a tangible connection between what is inside and what surrounds us. Imagine every complex feedback loop within these structures acting like a highly specific calibration tool, adjusting itself against some grander systemic standard we rarely measure. We are moving from observing internal upkeep to seeing how the cell maintains its relationship with totality. The practical truth here is that understanding this inherent self-organization provides a new metric for baseline wellness, a biological signature of true system equilibrium outside clinical failure markers. That process, mapping cellular integrity onto wider states, demands we recognize these mechanisms as active receivers across deep time.